Good afternoon. My name is Pratibha Rahman, and I'm a postdoc researcher at the Biomedical Informatics Department in Vanderbilt University's Medical Center. Today, I will be talking about our work on predicting brain metastases in non-small cell lung cancer patients. Up to 50% of advanced NSCLC patients develop brain mets. If we look at the figure on this slide, the x-axis shows days since lung cancer diagnosis, and the y-axis shows the percentage of patients still alive. The orange curve represents patients who develop brain mets within 24 months, and as we can see, it has a worse survival rate than the blue curve. Moreover, brain mets are not easily identified or predicted, leading to an unmet need in the care of NSCLC patients. So our goal is to identify clinical and genomic features associated with brain mets, as well as predict which patients will develop brain mets. Our data consists of NSCLC patients in Project Genie's Biopharma Consortium. The BPC consists of 4 academic institutions that have curated clinical data for patients who received NGS sequencing between 2015 to 2017. We exclude patients with brain mets at diagnosis as we're interested in predicting those who will develop them at a future time. Our covariates consist of demographics, anti-cancer therapies, and mutations and copy number alterations. The last 2 were restricted to oncogenic ones using OncoKB. We do univariate analysis with Fisher's test. We find that EGFR mutations are associated with an increased risk of brain mets, and this replicates findings of a previous study. We also find that nivolumab and alectinib are associated with a decreased risk, and this is interesting because both of these therapies penetrate the blood-brain barrier. For predicting the development of a brain met, we compare 5 different statistical models. Random forest uses majority voting from multiple decision trees. Support vector machines learn a high-dimensional boundary function for classification. Lasso regression uses L1 regularization, whereas ridge regression uses L2. And finally, the ensemble method uses average of ridge regression, SVMs, and random forests. We split our data into train and test sets with the 80 to 20 ratio. The training set is used by the models to learn parameters, whereas the test set is unseen. We use the area under the curve metric, which measures the discrimination of the model. So the higher the value, the better. We report the mean AUC with standard deviation of 10-fold cross-validation on the train set, as well as the AUC on the test set. Even though random forest and the ensemble method have the highest test AUC, we can see from the cross-validation results that there is no significant difference between the models. So to conclude, our models have reasonable prediction performance. However, one pitfall is that we lack radiotherapy data. Also, our current models do not account for the competing risk of death. This means that patients could die before developing a brain met, leading to misclassification. We're currently working on updating our models with landmark analysis to account for this. Thank you for your time. Please reach out via email if you have further questions.